ठीक है ठीक है we can start.
So Dr. Velazan, we are going to start now. Are you hearing my voice?
Sorry?
Are you getting my voice?
Yes, yes.
Okay. Because Dr. Nalatambi is unable to connect as a panelist. That's why I'll initiate. So we'll continue accordingly. Dr. Velazan, we are going to start now. Are you hearing my voice? Are you hearing my voice? Some noise is coming. One, two, three, four. Hello, 1, 2, 3, 4. Is my voice going? Hello. Mister. So, hello. So, Dr. Velazan, are you hearing my voice?
Yeah, it is clear. Yeah, please.
So, we are going to start now.
Yes.
So, good morning, everybody. So, this is the International Year of Plant Health 2020. So, to mark this, we are going to hold a series of webinars under the banner of Indian Phytopathological Society. So, dear IPS members, viewers, audience, and student friends, you know Indian Phytopathological Society is a third largest scientific society. And here, because of the ongoing COVID situation, so we have entered into another era. This is the virtual era. So I feel that this is like a historical breakthrough of our functionality. Because physically we cannot meet each other. That's why we have arranged this type of presentation, discussion, meeting. In future, till the COVID situation will continue. So our functions, our activities will go on like this in virtual mode only. So before instead of going for very long this introduction, so I would like to introduce our today's speaker. We are very much happy to see. So Dr. R. Velazan, he is from Oman. So let me read out his brief biodata very shortly so that we can save our time. Already we have lost 15 minutes. So he did PhD from Tamil Nadu Agricultural University in the year 1995. And he held various positions. Present in 2017 as associate professor in Sultan Qaboos University, Muscat, Oman. And also he has received many awards and honors, like most important one is the Rockefeller Foundation Rice Biotechnology Postdoctoral Fellowship he received in the year 1995 to 97. Then again, the Rockefeller Foundation Rice Biotechnology Career Fellowship in the year 2000 to 2003. Then Best Researcher Award from Tamil Nadu Agricultural University in the year 2004 and second time in the year 2011. And he received another award. This is Professor Hiralal Chakraborty Award from Indian Science Congress Association in the year 2003 and 4. And another one, that is Biotechnology Overseas Association Award from the Government of India in the year 2005 and 6. And he fellow of this present society, Indian Phytopathological Society, and he has published in peer-reviewed journals a large number of very high-rated research paper, almost more than 125 papers he has published, and he has written so far more than 10 book chapters. So I welcome again Dr. R. Velazahan. I'm very happy. Once again, I want to thank before you start your speech. So the platform is yours now.
Thank you very much. May I share the screen now?
Yes. Yes, we are able to see very clearly.
Okay, thank you. Respected Secretary, Indian Phytopathological Society, Dr. Robin Gogay. IPS South Zone President come Organizing Secretary, Dr. Nalathambi. IPS Southern Zone Zonal Councillor come Organizing Secretary, Dr. Uma Mageswari. And all the participants. Good morning to all of you. First of all, I'd like to thank Dr. Nalathambi and Dr. Robin Gogay for inviting me to give a presentation in IPS webinar series 2020. The topic of my presentation today is innovative strategies to reduce aflatoxin contamination in foods. The mycotoxins are toxic secondary metabolites produced by fungi, and most of these metabolites contaminate agriculture commodities and foods. Intake of mycotoxin contaminated food can result in toxicity in human beings as well as in animals. According to World Health Organization 2018, 25% of the world's food crops are affected by mycotoxins. The important genera of fungi which causes or produce mycotoxins are Aspergillus, Penicillium, Fusarium, and Alternaria. And the important mycotoxins are aflatoxins, fumonisins, ochratoxins, zearalenone, patulin, citinin, and deoxynivalenol. What are aflatoxins? These aflatoxins are toxic secondary metabolites produced mainly by two fungal pathogens. One is Aspergillus flavus, another one is Aspergillus parasiticus. These aflatoxins are carcinogenic, mutagenic, teratogenic, as well as immunosuppressive compounds. In this picture, you can see this growth of the fungus, that is greenish growth on the kernels as well as the maize cobs. This greenish growth represents the fungus, and the toxin produced by the fungus is called aflatoxins. The major susceptible crops are peanut as well as corn. In addition, the aflatoxin contamination is a major problem in chilies, sorghum, rice, almonds, and other tree nuts, including pista nuts and walnuts. There are more than 18 types of aflatoxins have been reported so far. For example, aflatoxin B1, B2, G1, G2, and aflatoxin M1. Among these aflatoxins, aflatoxin B1 is highly toxic. The fungus Aspergillus flavus produces two different types of aflatoxins, namely B1 and B2, whereas Aspergillus parasiticus produces B1, B2, G1, and G2. The toxigenic cells of Aspergillus flavus as well as parasiticus releases the aflatoxins through the process called exocytosis. So it has been proved by using anti-aflatoxin antibodies. You can see these fluorescent bodies on the mycelial walls of the fungus. So these are all the secreted points from which the aflatoxins are released into the medium. The International Agency for Research on Cancer classified the mycotoxins into different groups. The aflatoxin B1, B2, G1, and G2 comes under the group 1 that are carcinogenic for human beings. The aflatoxin M1 comes under the group 2B, which are carcinogenic for animal and potential carcinogenic for human beings. In addition, the ochratoxins as well as fumonisms comes under this category. The all other toxins, including deoxynivalenol, zearalenone, T2 toxins comes under group 3. The dietary intake of aflatoxin leads to the disease called aflatoxicosis in animals as well as in human beings. In poultry, it causes stunted growth, increased mortality, reduced exercise and production, liver and kidney damage, and suppression of immune system. In the case of dairy cattle, it causes reduced feed intake and weight loss, decreased reproductive performance, liver and kidney damage, and depression of immune system, and edema in the abdominal cavity. In case of human beings, it causes liver cancer. In addition, it suppresses the immune system. This picture shows the biotransformation pathways of aflatoxin B1. In the body, if we consume this aflatoxin contaminated food, due to the enzyme cytochrome P450 enzyme, the aflatoxin B1 is converted into highly reactive group, that is aflatoxin epoxide. So this highly reactive group, it will form or combine with the DNA and form DNA adducts, and then that causes the DNA damage. Finally, that will result in cancer. In addition, the epoxide form bind with the protein and induces a toxicity. The symptoms related to the exposure to aflatoxins depends on the type of toxins, which type of toxin, whether it is aflatoxin B1, G1, or G2 or B2, and the levels of aflatoxins in the food. What is the quantity of aflatoxin present in the foods? Then amount of such contaminated food consumed, then body weight of the individual, and physiological state of the individual. This picture shows the presence of aflatoxin in the food chain. Normally, the human beings are exposed to aflatoxin through direct consumption of contaminated food. If the contaminated materials are not fit for human consumption, normally our tendency is to put it for human or animal feed. So when it is mixed with the animal feed, when the animal consumes such kind of contaminated feed materials, we can expect the presence of aflatoxins in the meat, egg, as well as in the milk. It will enter into the food chain. So the presence of aflatoxins in the food chain threatens the people's livelihood, food security, as well as human health. Several studies shows the presence of aflatoxins in the meat of broiler chicken that were fed with aflatoxin contaminated feed. So the feed to egg aflatoxin transmission ratio is up to 0.8%. Similarly, when the milk animal consume aflatoxin contaminated feed, the aflatoxin B1 is converted into M1 in its animal body and secreted in the milk. And the conversion rate is up to 6.2%. So this table shows the effect of different levels of aflatoxin consumption on human and animal health. Up to 20 parts per billion in the food, there will not be any health hazard to human beings. Up to 50 parts per billion, no health hazards in animals. Whereas above 400 parts per billion causes liver damage in both human beings as well as in animals. So the US Food and Drug Administration has set an aflatoxin tolerance limit of 20 parts per billion for foods, feeds, as well as for feed ingredients. According to World Health Organization and FAO regulation, the content of aflatoxin M1, which is present in the milk, should be less than 0.5 microgram per liter. The dietary exposure can vary greatly from country to country. According to the World Health Organization report 2018, the developed countries are exposed to 1 nanogram per kilogram body weight per day, whereas African countries, they indicate above 100 nanogram per kilogram of body weight per day. According to the World Health Organization report in 2015, the aflatoxin caused 22,000 illness and 20,000 death worldwide. This is the worldwide figure. In the particular year 2015, it caused 22,000 illness and 20,000 death. The aflatoxin contamination causes economic losses to the producer, government, and consumers. For producer, it causes less income because of the contaminated food commodities, then reduced productivity of livestock, increased storage, transportation, and packaging cost, and increased cost due to surveillance and control. For government, the aflatoxin contamination lower the foreign exchange because of reduced exports, and increased cost due to sampling and analysis, and increased expenditure in human health and livestock care services, and increased cost of training and extension programs. For consumers, it causes impaired health, then possible higher medical and veterinary costs. And we conducted a survey in different parts of Tamil Nadu when I was working at Tamil Nadu Agriculture University. And we collected samples of pre-harvest maize kernels and post-harvest maize kernels, food samples, poultry feeds, and livestock feeds, and analyzed for the presence of aflatoxins. And we observed 32% of the pre-harvest maize kernels and 22% of the post-harvest maize kernels and 12% of livestock feeds contain above tolerance level fixed by FDA. The aflatoxins are extremely durable under most conditions of storage, handling, and processing. Several studies indicated that heat treatment cannot reduce the aflatoxins. For example, this study shows the effect of heat treatment on reduction of aflatoxin in wheat. The others used different temperature and different duration. The results indicated that the level of aflatoxin B1 in wheat reduced to 90% by heating at 200 degrees Celsius. So even after heating at 200 degrees Celsius, the aflatoxin cannot be removed completely. Even 10% of the aflatoxins still remains in the commodities. The another study was conducted to study the effect of roasting on aflatoxin in pista nut. The others used a different temperature, 90 degrees Celsius, 120 and 150 degrees Celsius, and different durations. The results indicated that roasting at 150 degrees Celsius for 2 hours degraded more than 95% of the aflatoxin in pista nuts. However, the resulted product was not edible. So mere presence of the toxin producing fungus on the surface of the kernels will not automatically indicate the presence of the toxins, because many factors are involved, because some of the aspergillus flavors are non-toxic, so we cannot expect the presence of the toxin always. Similarly, the absence of visible mold does not indicate that the commodity is free from aflatoxin contamination, because the mold can be removed by polishing. Still, the toxin will be present in the commodity. So the analysis of sampling, sampling is the most important contributor to reduce the variability. Proper sampling should be used for analysis. So the errors of sampling are expressed as seller's risk and buyer's risk. The seller's risk means the risk of rejection of good batch of commodity because of poor sampling. Similarly, the buyer's risk is the risk associated with the acceptance of a bad batch. So the sampling is important. The variability can be minimized through increasing the size of samples as well as the subsamples. According to the European Commission, if the container contains more than 50 tons of cereal grains, minimum 100 subsamples should be drawn for analysis, and the total weight of these subsamples should be approximately 10 kilograms. If the consignment is less than 50 tons, 10 to 100 subsamples should be drawn, and the total weight should be 1 to 10 kilograms. If it is less than 0.5 tons, the number of subsample may be less, but the total weight should be at least 1 kilogram used for analysis. Regarding the methods of analysis of aflatoxin, there are two different methods. One is a rapid method, which includes the ELISA and dipstick assay kit. And the routine methods are thin layer chromatography, high performance liquid chromatography, and LCMS fluorometer. These are all routine methods. Each method has its own detection limits, and these ELISA kits can detect up to 1 parts per billion, whereas the dipstick assay kit can detect 4 parts per billion. And the TLC method is qualitative, and HPLC method is widely used in several laboratories, and the detection limit is 0.05 parts per billion. So the HPLC method, LCMS and ELISA are nowadays used in different laboratories, and the HPLC method is highly sensitive. It takes a very short time for analysis, and automated, there is auto sampler facility is available. And ELISA method is convenient and sensitive and rapid, and it uses less of organic solvents for extraction. So these two methods, HPLC as well as ELISA methods, are widely used in several laboratories. And LCMS is highly sophisticated technique, and it has high sensitivity, and we can analyze many number of toxins at the same time. Moreover, structural information of the analyte can be obtained by using LCMS. And which method can be used for analysis? It depends on the purpose. If it is for regulatory application, for example, if we are planning to send, if it is for export, we need to adapt the method recommended by WTO. The acceptable methods may vary between countries. So the standard methods prescribed by CEN as well as AOSC should be adapted if it is meant for export. So the aflatoxin contamination can occur at any stage of the food supply chain. It may be during the pre-harvest stage or in storage, that is post-harvest. So we have to adapt several management strategies to reduce aflatoxin contamination in agriculture commodities. At the pre-harvest stage, some of the strategies like growing resistant or tolerant varieties, or following good agronomical practices and biological control, biotechnological approaches can be used at the pre-harvest stage. After harvest, the strategies like reducing the moisture content to 8%, then storage of the harvested produce under clean, dry, and insect-free conditions, and treatment of the commodities with the electromagnetic radiation like gamma radiation or UV radiation, and ozone fumigation, improved packaging methods, improved sorting technologies, and detoxification. Here I would like to give some information about the research conducted on biological control, biotechnological approaches, and improved sorting technologies and detoxification in this lecture. Regarding biological control, some products, commercial products, are already available in the market in other countries. For example, Aflatoxin. So this biocontrol product based on Aspergillus, that is non-toxigenic strain of Aspergillus flavors, it is available, and it is marketed by Syngenta company in US. This is mainly used in groundnut field for the control of aflatoxin contamination in groundnut. So this Aflatoxin is a non-toxigenic strain, and it reduces the aflatoxin contamination up to 90%, mainly through the competitive exclusion of the toxigenic strain. The IATA released another product, namely Aflasave, mainly for the use in maize as well as in groundnut for reducing the aflatoxin contamination. So we tried to exploit this biological control for the management of aflatoxin contamination in groundnut. We screened several bacteria. For the antagonistic activity against Aspergillus flavors, and we found one soil bacteria which can reduce this or which can inhibit the growth of Aspergillus flavors. This is control plate. This one is treated with the same strain in 3 different places. We can see very high level of inhibition of the growth of Aspergillus flavors. So this strain was subsequently used for testing for its effect on seed germination and seedling vigor of groundnut. And we developed a formulation, talc-based formulation of this bacterial strain. This bacterial strain was identified as Burkholderia based on the biochemical test as well as by 16S RNA sequence analysis. Upon treatment of seeds with this Burkholderia, we could see, so we observed the increase in seed germination, shoot length, root length, as well as seedling vigor. So then we studied the effect of various medicinal plants for the inhibitory activity against Aspergillus flavus. And we observed a medicinal plant, namely Zimu. That's a natural hybrid of onion and garlic. And this plant has antifungal activity against Aspergillus flavus. And this picture shows the inhibition of Aspergillus flavus by Zimu extract. When the groundnut seeds were dipped in the Zimu extract, it can inhibit the growth of Aspergillus flavus. In addition, the root exudates of Zimu, it has a detoxification potential, aflatoxin detoxification potential. It was observed by a small experiment. The roots of Zimu plants were dipped in aflatoxin solution and incubated for 5 days at room temperature. Then the applied aflatoxin was recovered by extraction with the chloroform and it was analyzed. And the results indicated that the root exudates of Zimu plant can degrade aflatoxins. So this arrow indicates the position of B1. When the roots of Zimu was dipped in the aflatoxin solution, we can see the reduction in the aflatoxin content. Then we conducted an experiment to incorporate both Zimu intercropping as well as application of biocontrol agents, namely Burkholderia, for the management of aflatoxin contamination in groundnuts. And these are the different treatments we used. One is Zimu intercropping, that is 1 row of Zimu and 4 rows of groundnut, and then seed treatment with the Burkholderia, and soil application with the Burkholderia at different days after sowing, then seed treatment plus soil application, and so on. And the results indicated that the seed treatment and soil application with the Burkholderia significantly reduced the aflatoxin content and increased the pod yield. In addition, transgenic strategies can be used to reduce the aflatoxin contamination in peanut as well as in corn. There are 3 basic strategies for reducing utilization of the transgenic resistance, namely reducing infection by the pathogen. That is one strategy. The another strategy is inserting genes capable of degrading the toxin. The third one, reducing the mycotoxin accumulation by interfering with the biosynthetic pathway. So these 3 approaches can be used to reduce the aflatoxin contamination. This author Rarang and co-workers recently reported the host-induced gene silencing to reduce the aflatoxin contamination in transgenic maize. So in the aflatoxin biosynthesis, there are several genes involved, and these authors use the AFL-M gene that is coding for versicolorine dehydrogenase. So this is one of the important enzymes which leads to the synthesis of aflatoxin in Aspergillus flavus as well as in Aspergillus parasiticus. So these authors targeted this AFL-M gene, which is coding for versicolorine dehydrogenase. By employing the gateway cloning system, they prepared the RNA-I vector. After construction of this RNA-I vector, the maize inbred lines were transformed with this construct and the transgenic plants were obtained. This is the flow chart showing the construction scheme of the versicolorine gene. And finally, this construct was under the control of entire cassette is under the control of 35S promoter. And by using agrobacterium-mediated transformation, the transgenic plants were developed. And these transgenic plants were tested in T1 as well as in T2 generations. And some of the transgenic plants showed significant reduction in the aflatoxin contamination in the kernels. This assay was based on kernel screening assay. Artificially, the kernels were inoculated with aspergillus flavus, and then aflatoxin was estimated after sufficient duration, and they could observe significant reduction in the aflatoxin contamination in the kernels. Then they observed under field conditions, and there was no phenotypic difference in the height of the plant or a number of kernels per year. And phenotypically, there was no difference among the transgenic plants as well as the non-transgenic plants, but the transgenic plants showed lower level of aflatoxin accumulation in the kernels. In another study, the same gene silencing, host-induced gene silencing method was used to develop transgenic maize plant. However, here in this case, the target gene was AFLC gene, another gene. So this is the total, the size of the AFLC gene is around 6,594 base pair, and they selected 3 regions, each consisting of 200 base pair, and they made a construct, and these 3 regions were arranged in tandem with this spacer, and the inverted repeat construct was used, and this entire cassette was under the control of the endosperm specific promoter. So here they wanted to express only in the endosperm, so they used endosperm specific promoter, that is gene promoter, and they used agrobacterium mediated transformation to develop transgenic maize plant. And these maize plants were assayed for, since it has a bar gene selection, they used glufosinate ammonium solution for testing of these plants, and the null plants showed the toxicity, because upon the leaf painting assay, the non-transgenic plants showed toxicity, whereas the transgenic plants were resistant to the glufosinate. So they tested the transgenic plants by artificial inoculation to Aspergillus flavus, and this shows, this black portion shows the site of inoculation, and they tested these grains around this site of inoculation for analysis, and they observed that the transgenic plants were completely resistant to aflatoxin contamination, whereas the non-transgenic plants showed higher level of aflatoxin accumulation. The transgenic plants were completely free from aflatoxin contamination. In another study, the Williams and the co-workers, they studied the effectiveness of the transgenic corn hybrids expressing Cry1AB gene and studied the aflatoxin accumulation in such kind of transgenic plants. So the southwestern corn borer is a major problem in corn. It feeds on the corn and makes injuries. These injuries will be helpful for the fungus, that is Aspergillus flavus, to enter and then colonize these maize kernels. So they studied the effect of Bt gene on these aflatoxin accumulation, and they observed that Bt hybrids were completely resistant to the aflatoxin accumulation. And this table shows the difference between non-Bt plants as well as the Bt plant. They could observe significant reduction in the aflatoxin content in the maize kernels. Regarding the post-harvest management strategies, improved sorting techniques can be used, and detoxification method can be used for degradation of aflatoxins. Wang and co-workers in the year 2014, they standardized a method for sorting of the infected grains. They used near-infrared spectral imaging technique for detection of aflatoxins in maize kernels. These methods are fast and inexpensive, moreover non-destructive, especially when we are going for analysis of pista nut, the cost of pista nut. So without the destruction of the pista nuts, we can analyze the presence of the aflatoxins in the harvested material, harvested produce. And it requires little sample preparation. And this imaging techniques uses the wavelength between 1000 to 2500 nanometer, and it can detect aflatoxin B1 up to 10 parts per billion. So after this imaging technique, they analyzed by chemical methods also, they confirmed the results. So this technique can be used for sorting of the kernels. It can be used for both maize as well as peanut. The next one is the detoxification of aflatoxins. So once the aflatoxin is formed, it is very difficult to eradicate. So we can use the method of detoxification of aflatoxin. And this detoxification method should follow certain norms, and the toxin should be destroyed or transformed to non-toxic compounds. The food or feed material should retain its nutritive value after treatment. That is more important. And the physical properties of the material should not be altered. And the detoxification process should be economically feasible. In the case of detoxification of aflatoxin in feeds, we can use different methods like ammoniation, addition of adsorbents, and biological detoxification. The ammoniation is an approved method for detoxification, but the drawback is there are possibility of degradation of animal health by residual ammonia in the feed. Then some adsorbents like toxin binders are available in the market. However, this was not approved by FDA. So these toxin binders are natural clay particles. These clay particles like bentonite or zeolite or hydrated sodium, calcium, aluminum silicates, these clay particles are mixed with the feed, and then it can be used for feeding animals, especially the poultry birds. So these clay particles bind with the toxin which are present in the feeds, and then it will come in the excreta, and it not allow the aflatoxin to dissociate in the gastrointestinal tract. So all this aflatoxin bound material will be come in the excreta. This is commercially available and many countries are using it. And this commercial name is Novosil Plus, and it is used at a dose of 0.1 to 0.5%. In addition, certain biological agents can be used for detoxification process. So the drawback is the utilization of nutrients from food. For example, lactic acid bacteria can be used for the biological detoxification of aflatoxins. This lactic acid bacteria, when it is mixed with the feed, it will utilize some nutrients for its own growth. In addition, it secretes some undesirable compounds in the media. So we cannot use such kind of toxin binders for foods. So for foods, for detoxification of aflatoxins, some physical methods, chemical methods, and microorganisms are being used. The physical methods like heating and roasting can be used. However, certain nutrients are destroyed during this process. Some chemical agents like citric acid, then sodium hydrosulfide, and isothiocyanates are also being used for detoxification of aflatoxin. However, their applicability in food is restricted because of the safety problems and chemical residues. So similar to the feed, the microorganism can be used for detoxification. For example, lactic acid bacteria as well as yeast like Saccharomyces cerevisiae. However, these organisms will take some nutrients from the food materials. In addition, they may secrete some undesirable compounds. So what is the alternative? We tried to exploit the medicinal plant extracts for detoxification of aflatoxins. So we screened several medicinal plants belonging to different families for detoxification of aflatoxins, and we used this strategy. The aflatoxin was mixed with the plant extract and incubated for 48 hours at 37 degrees Celsius, and the applied aflatoxin was re-extracted by using chloroform, and the content of aflatoxin was analyzed by TLC or ELISA technique. Finally, we identified two medicinal plants. One is Ardadada vesica, another one is Trachysperma mummi, that is mummy seeds. So the extracts derived from these medicinal plants have the potential to degrade aflatoxins. This picture shows the detoxification of aflatoxins. So this lane shows the aflatoxin B1 control. When it was mixed with the Trachysperma mummi extract, you can see the drastic reduction in the fluorescent compound. Similarly, the B2 also shows reduction, whereas G1 complete absence. We can observe complete degradation of aflatoxin G1 by mixing it with the Trachyspermum mummi extract. So G2 also shows the reduction. So these Trachyspermum mummi seed extracts can detoxify aflatoxins. Further, we studied whether dialysis can remove its activity. Similarly, we studied the effect of boiling or heat treatment on detoxification potential of aflatoxin. Here we used aflatoxin G1 for its for testing, and we observed boiling can reduce the degradation potential of mummy extract. Similarly, dialyzed plant extract dialysis has no effect on the detoxification potential. Even after dialysis, the extract can show detoxification. That indicates the compounds are high molecular weight because the crude extract showed 64.5% degradation. After boiling, it was reduced to 44.8%, whereas the dialysis, by using dialysis membrane with the molecular cutoff of 12 to 14 kilodalton, showed 91.4% reduction in the aflatoxin. So that means that it has a high the molecular weight of the compounds present in the Trachyspermum mummi extract is of high molecular weight. Further, the time course study indicated that up to after 6 hours of incubation, it observed 78% reduction or detoxification, and after 12 hours, it showed 88%. After 24 hours of mixing, we could observe above 90% degradation of aflatoxin. Then we studied the structural properties of the degraded product. So we compared with aflatoxin. What happened to the aflatoxin chemical after treatment with this Trachyspermum mummi extract? So this is the aflatoxin standard. So we could see a molecular ion peak at 329, which is specific for aflatoxin G1. Whereas after treatment with the Trachyspermum mummi extract, we could see a major ion peak at 288. Then we use this molecular ion peak for further processing, and MS analysis of this precursor ion at 288, we could observe a breakdown product of at 270. So this product, so based on this, we predicted the molecular structure of this degradation product. And this is the structure of the aflatoxin G1. It has a bisfuran ring as well as a lactone ring. And the metabolites at 288 as well as 270 are showing this structure. So it indicates the modification of the lactone ring structure. So any modification in the lactone ring structure may result in the loss of toxic properties. So upon treatment with the Trachyspermum mummi extract, it changes the lactone ring structure, which results in less toxic of the toxin compounds. The another test is chromosomal aberration test. The aflatoxin G1 can induce chromosomal aberration in maize, especially at the anaphase stage. The samples can show degradation, that is chromosomal aberrations. You can see this breakdown product. Whereas when the aflatoxin G1 was incubated with the Trachyspermum mummi extract, the aflatoxin could not induce any such aberrations on the chromosomes. So that indicates this is another evidence for the degradation of aflatoxins by the Trachyspermum mummi extract. So another plant that showed a detoxification potential is Adadora vesica. This is commonly available everywhere, but the leaf extracts of Adadora vesica show detoxification potential. And this table shows the degradation of aflatoxin, all 4 aflatoxin, B1, B2, G1, and G2. It showed more than the 88% of detoxification of all 4 aflatoxins. And this is the time course study of aflatoxin B1 detoxification by Adadora vesica extract. After 48 hours, more than 90% reduction or degradation of aflatoxin was observed. And the HPLC analysis showed this is aflatoxin B1. This is the peak responsible. This is the peak for aflatoxin B1. After mixing it with Adadora vesica extract, that peak could not be detected. So that indicate that complete degradation of aflatoxin B1 by Adadara vesica extract. Further, the mass spectrometry analysis indicated that the peak fraction for aflatoxin B1, that is 313 mass to charge ratio, could not be observed after treatment with the Adadara vesica extract. So, however, the B1 standard showed this is peak, that is 313, whereas it was completely absent after treatment with the Adadara vesica extract. So that indicates complete degradation of degradation or detoxification of aflatoxins. Then we wanted to study the chemical nature of the material present in the Adadara vesica extract. So we tested for phenolics, terpenoids, and alkalides. Finally, we observed the reaction of Adadara vesica extract for alkalides. So this is the TLC showing the presence of 3 different alkalides. That is RF value 0.37, 0.47, and 0.63. And we removed or scratched out each band separately. And it was each band was tested for detoxification potential. And we observed the fraction number 3, that is RF value with the 0.37, showed high levels of detoxification of aflatoxin. And based on the analysis by GCMS, we observed that compound matches with quinoxaline. So it is an alkaloid. So the results indicated that the alkaloids present in the Adadara vesica extract is responsible for degradation of aflatoxins. What are the constraints for the adoption of aflatoxin management options? First one is lack of awareness and knowledge among farmers, traders, processors, and consumers. Then aflatoxin infection has not been attributed to cause yield loss. There is no reduction in the yield. So the farmers need not worry about aflatoxin, aspergillus infection or aflatoxin contamination. Then aflatoxin may be present in the food without any noticeable effect on taste, smell, or appearance. There are no visible marks of degradation of quality. Except the mycelial growth, we cannot observe any kind of degradation of quality. And there is no premium price for aflatoxin-free produce. So even if farmers follow all measures to produce aflatoxin-free commodities, there is no premium price. In conclusion, you can say the most commonly occurring aflatoxin in foods are aflatoxin B1, B2, G1, G2, and M1. Among the aflatoxin B1 is the most potent carcinogenic compound that induces hepatocellular carcinoma in human beings. The Trachisperma mummi and Adadara vesica extracts can be used as a biologically active alternative to detoxify aflatoxin, and it has a huge potential to be used in food and feed industries. And biological control in combination with good agricultural and storage practices, as well as efficient post-harvest management, should be followed to reduce the aflatoxin contamination. The maize lines resistant to aspergillus flavors may be developed through genome editing tools. There are many recent reports indicate that there are some susceptibility genes for aspergillus flavors infection have been identified. So these genes can be knocked out by using the CRISPR-Cas9 and other genome editing tools to develop a transgenic plants. And stringent enforcement of regulations and advanced sorting and testing procedures are necessary. And the last one is rejection should not re-enter the food chain. That is more important. If the consignment is rejected for export, it will come to the local market. That should be avoided. I'd like to acknowledge Sultan Qaboos University, Oman, where I am currently working, the Research Council of Oman for funding for aflatoxin detoxification project, and Tamil Nadu Agriculture University, Coimbatore, Kansas State University, USA, ICAR, and DST, Government of India, and Department of Biotechnology, Government of India. And finally, I need to thank all my students, senior research fellows, and research assistants worked under my supervision. Thank you very much.
So, very nice talk we have heard just now from Dr. Velajahan. So we are very much thankful to you. Despite of your busy schedule, you have spared some time for our society. That's why I once again thanking you. But so far, to save our time, we have received total 17 questions. Now this is the question answer session. But instead of going each and every questions from these participants, so selectively one or two questions I'll be feeling to have discussion here. So here out of 17, 18 questions, so whatever the questions will be remaining from the participants, so we'll be providing the direct link and email ID of Dr. Belajahan so that they can discuss directly with him. So now let me pick up one or two questions so that we can discuss here itself. Here one question is there, sir, how to manage the aflatoxin contamination by bioproducts?
Okay.
How to manage the aflatoxin contamination by bioproducts?
Okay. So we can use the biological control agents at the pre-harvest stage. And at post-harvest stage, as I mentioned in my lecture, we can use some of the plant products, then biological agents for mixing with these contaminated materials. So for example, lactic acid bacteria. So these lactic acid bacteria physically bind with the toxins. The disadvantage, as I mentioned here, it will absorb some nutrients from the host. So we need to find out an alternative strategy. That's why we selected the medicinal plants. So this medicinal plant, that is a powder or extract derived from such kind of medicinal plants, can be mixed with this product so that it can degrade the aflatoxins.
Okay, thank you. So our this participant's next question is, so Amita, he's asking, normally pathogens of plants are non-pathogenic in humans. Why are aflatoxins affecting humans? I repeat, normally pathogens of plants are non-pathogenic in humans. Why are aflatoxins affecting humans?
Okay. So the aflatoxins are metabolites of the fungi. So once if we consume aflatoxin contaminated food materials, it forms, as I told you, that because of this enzyme cytochrome P450 enzymes, the epoxide form is formed. So the epoxide form of aflatoxin is more dangerous. It forms adducts, it joins with the DNA and interferes with the DNA replication as well as the transcription as well as the translation process. That finally leads to the cancer. The toxin after formation of epoxide, it is highly dangerous.
Okay, thank you. So the last questions I would like to put here, this is from Shivam. So he's asking, what are the congenial conditions required for production of aflatoxin in maize? I repeat, what are the congenial conditions required for production of aflatoxin in maize?
Okay. Regarding the temperature, even 20 degrees Celsius to 40 degrees Celsius, the aspergillus can grow and produce aflatoxins. But in the maize, mainly the aspergillus flavus enters through the silks. And moreover, injuries by insects will pave entry for the aspergillus flavus. So that during cultivation, there should not be any mechanical injuries. Even, for example, birds injuries will form sign. Through these injuries, the aspergillus flavus can enter, because aspergillus flavus and parasiticus is present in throughout, say, even it is present in the air, soil, and water. So these aspergillus can enter through these wounds. So the wound should be avoided. That's why the transgenic plants which are expressing BT can reduce the insect infestation. So because of if it reduces the boreholes, the entry can be avoided. So good agriculture practices should be followed to reduce the aflatoxin contamination in maize.
Thank you, Dr. Velajan. So we want to stop here question session. So far we have received 30 questions. Out of these 30 questions, you have very nicely and convincingly answered 3 questions. And I will request all the these participants who is giving their questions. I think they will get the email ID of our speaker, the Dr. R. Velajan, in the this webinar schedule. So you may directly put your question to him to get the best answer from him directly. So thank you. So now I'm passing on my desk to our treasurer, Dr. Malkhan Singh, to place vote of thanks.
So, in formal vote of thanks, I would like to thank our society president, Dr. P.K. Chakravarti sir, who guided us to organize this IPS webinar series, and chief editor, Dr. B.N. Chakravarti, then our secretary, Dr. Robin Gogoi, and joint secretary, Dr. Kalyan Mandal, all the executive members, including journal president and councillors, and special thank to Dr. Velajan, who gave the wonderful talk, including aflatoxin, detection, detoxifications, and diagnostic assays. That is very useful to our for scientists and other students. And thanks to Dr. Nala Thambi and Dr. Uma Maheshwari Madam, who organized to conduct this IPS webinar series in Southern Zone, and including all the scientists and students and IPS members who joined this this IPS webinar series, and all the participants, and including our office bearer, Mr. Raju, Sipra, and Vishal. Thank you, one and all.
See you in future again. Yeah. Thank you.
Thank you, one and all.